to a day in the genetics lab. Today, I'll walk you through the full journey of identifying a mysterious yeast mutant, CDCX, using transformation rescue and recombinant DNA cloning. This is a real story from our semester-long investigation. Yeast cells normally divide through budding, but our CDCX mutant behaves strangely. At the permissive temperature, 25 degrees Celsius, everything looks normal. But shift the temperature to 37 degrees Celsius, the restrictive temperature, cell division starts to collapse. The buds elongate, cytokinesis fails, and the yeast form long chains of incompletely separated cells. This tells us that the mutation affects one of the four septin genes, CDC3, CDC10, CDC11, or CDC12. They are proteins essential for forming the septin ring at the mother bud neck. When septins malfunction, the entire architecture of division collapses. This mutation is temperature sensitive. The protein works at low temperature but misfolds at high temperature. This is a classic conditional allele where heat destabilizes the protein. Our mission for the entire module is twofold. Number one, identify which septin gene is mutated using a transformation rescue assay in yeast. And number two, clone and sequence the mutant allele using recombinant DNA methods to understand the exact DNA change. Let's start with the rescue experiment. To determine which gene is broken, we perform a transformation rescue. Each student transforms the CDCX strain with one plasmid carrying a wild-type copy of CDC3, CDC10, CDC11, or CDC12. These plasmids also contain URA3, allowing only successfully transformed cells to grow on URA plates. I was given the plasmid that encodes the gene CDC11. Others in the class received one of the other three plasmids. Together, we would be able to see whose plasmid was successful by mixing the competent CDCX cells, that is, the cells that have been chemically treated to increase the DNA uptake, with our plasmid and incubate them with the lithium acetate SS-DNA back transformation mix, called solution 3 in our handout. Lithium acetate neutralizes the negative charge on DNA membrane, while single-stranded cell and sperm DNA protects the plasmid and acts as a carrier. BEG brings the DNA close to the cell surface and promotes uptake. After adding the plasmid, we slowly add 500 microliter of solution 3 and vortex vigorously to mix. This is where the long incubation happens. We incubate it for an hour, and during that hour we literally placed the tube in the 30 degrees Celsius water bath, vortexed it every 15 minutes to keep the DNA, BEG, and cells well mixed. This step dramatically boosts our transformation efficiency. After the 1 hour incubation, we plate 100 microliter of our transformation into two YED plates, that is yeast extract dextrose plates, which were then incubated at permissive temperature and restrictive temperatures. If the plasmid rescues CDCX, colonies should grow normally at 37 degrees Celsius. If not, the cells would fail to septate, and thus would not be able to grow, and we wouldn't be able to see the colonies form on the YED agar. And here's what we see. No growth at restrictive temperature for the CDCX cells transformed with CDC11. What a bummer. At least now we know that CDC11 does not rescue our cells, so we can rule out that CDC11 was a gene mutated in our CDCX cells. By the end of this experiment, with other students, we can determine which septin gene is defective, based on whether the wild-type gene complements the mutant phenotype. It turns out to be CDC3.
A new record. Reset it!
Once we know which gene is mutated, the next challenge is to identify the exact DNA mutation. In order to do so, we need to PCR amplify the mutant allele, ligate it into a cloning vector, transform the recombinant plasmid into bacteria, select for successful transformants, and finally, perform restriction digestion and gel electrophoresis to verify that our insert is present. Before lab, Dr. Hartman PCR amplified the mutant gene directly from the CDCX strain. Taq polymerase adds an extra adenine nucleotide to the 3' end of PCR products. This creates an A overhang. The PCR4 Tobo vector, provided in a commercially available kit, has complementary T overhangs and is pre-bound to the topoisomerase 1, an enzyme that ligates the insert without restriction enzymes or ligase. This is called Topo-TA cloning. Once the insert is ligated, we transform the recombinant plasmid into chemically competent E. coli. selection is done on ampicillin and X-gal plates. Ampicillin is a normal antibiotic selection mechanism where only cells with our recombinant DNA will survive, while regarding X-gal, if the plasmid did not receive our insert, LAX-Z gene remains intact. The colonies turn blue when they metabolize X-gal. If the plasmid does contain our insert, the insert disrupts LAX-Z, preventing beta-galactosidase production, and then the colonies remain white. Finally, we perform mini-prep, ECOR1 digestion, and gel electrophoresis to confirm that our plasmid contains the 1.5 kilobase mutant insert alongside the 3.9 kilobase vector backbone.
Copy that. Initiating phase 3. Let's hope they hear this.
Now let's walk through the recombinant DNA workflow with the actual methodology that we used in lab. First, we add the PCR amplified mutant CDCX gene to the topo vector. As soon as the AT pairing occurs, the topoisomerase ligate them together in seconds. We start by setting up the topo TA cloning reaction. Into a sterile PCR tube, we combine 2 microliter of the PCR amplified mutant gene, 1 microliter of the provided salt solution, 1 microliter of the PCR for topo vector, and 2 microliter of nucleus-free water. We mix the reaction gently and incubate the tube for 5 minutes at room temperature, then immediately place it on ice to preserve the ligated plasmid. Next, we perform the chemical transformation using top 10 E. coli, using the calcium chloride heat shock method. These cells remain on ice for at least 10 minutes before use because the cold stabilizes their membrane and prepares them for DNA uptake. We add the 2 microliter of our turbo reaction directly into 50 microliter of ice-cold content cells and flick gently to mix. The tube stays on ice for 30 minutes, allowing the plasmid DNA to associate it with a chilled bacterial membrane. Then comes the heat shock step. We transform the cells from ice to a 42 degrees Celsius water bath for exactly 30 seconds. This brief heat pulse forms transient pores in the membranes and drives plasmid into the cells. Immediately afterwards, we return the cells to ice for 2 minutes to help the membrane reseal. To allow the bacteria to recover, we add 250 microliter of form SOC medium and incubate the cells at 37 degrees for 1 hour. Shaking gently, we help the cells express the ampicillin-resistant gene. Once recovery is complete, we plate 10 microliter and 100 microliter of the culture onto LP plates containing ampicillin and X-gal. We incubate it overnight, and there's the culture. These overnight colonies are used for mini-prep SDS, and sodium hydroxide lysed the cells and denatured DNA. This is in buffer P2. Neutralization precipitates chromosomal DNA while plasmids rapidly reanneal and stay in the solution. This occurs in buffer N3. Spin colons purify plasmid DNA effectively. To verify that cloning succeeded, we set up an ECOR1 restriction digest. ECOR1 cuts at the 2 engineered sites flanking the topo insertion region, releasing our 1.5 kilobase mutant insert from the 3.9 kilobase PCR4 vector backbone. After running the digest on agar gel, successful cloning is indicated by 2 clear bands, the 3.9 kilobase vector and the 1.5 kilobase insert. Seeing those bands confirms that our recombinant plasmid contains the correct mutant gene and is ready for sequencing.
Really necessary?
Yes, it is, okay? This is a groundbreaking scientific discovery. We can't just bury it like some common mammal, okay? It's not a dog.
All right.
By combining classical yeast genetics with modern molecular cloning, we could uncover both the gene responsible for the CDCX mutant phenotype and the exact DNA mutation that causes the temperature-sensitive defect. This project shows how geneticists bridge phenotype to genotype, starting from a strange mutant under the microscope and ending with a sequenced allele in hand. Thanks for watching and welcome to a typical day in the genetics lab.